Recording. So, hi everyone, and thank you for joining in for the Synthetic Biology Club hosted by the Abdel-Mawgoud Synthetic Biology Lab. As, this monthly online event aims to promote discussions around synthetic biology and also to share latest scientific advances on the subject. And as always, the format of the event includes a presentation of up to 40 minutes, followed by a period of questions and discussions of up to 10 minutes. And before we start, I would like to remind everyone to keep their microphone turned off in respect for the presenter. And also, I would ask you to keep your questions for the end, or if you think that you will not remember them, you can write directly your questions in the chat and we'll go by the questions at the end. And today's presenter who kindly accepted the invitation is Adegbaju from the Institute of Plant Biotechnology at Stellenbosch University of South Africa and from the Department of Crop Production and the Federal University of Technology, Akure of Nigeria. He'll be discussing with us on his own research work titled Analysis of Interaction between glucan, water dikinase, and starch benching enzyme in determination of starch structure. And just to give you a bit of information on our presenter, Adegbaju Muyiwa Sei is a doctoral candidate under the supervision of Professor Lloyd at the Institute of Plant Biotechnology in Stellenbosch University, and supported by the NRF TWAS African Renaissance Doctoral Scholarship for Research in South Africa. His doctoral research, now awaiting final oral presentation, is aimed at elucidating functional interaction between some starch metabolic enzymes in Solanum tuberosum, and he was awarded a Master in Agricultural Technology in 2015 and a Bachelor in the same field in 2010, all in the Department of Crop, Soil, and Pest Management at the Federal University of Technology, Akure of Nigeria, where he has also worked as a teaching assistant from April 2014 to December 15. In January 2016, he accepted a position of assistant lecturer at the department where he obtained his first and second degree and taught courses on genetics and biotechnology prior to commencement of his study at the Stellenbosch University in March 2018. He's currently an enthusiast in bioeconomy and thoroughly enjoys research in this area. Now I'm done with talking and I leave it to you, Muyiwa.
Okay, thank you very much. Thank you for the introduction. Please let me share my screen with you. Okay, can you hear me clearly? Just want to be sure.
Yeah, we can see your presentation.
Okay, all right. Thank you very much. So thank you for the introduction. I'm Muyiwa by name, and the supervisor of my project is Professor James Lloyd, and the co-supervisor's name is there written. And the title of my project, actually, this is just one part of the project, of my PhD project. So the title is Analysis of the Interaction between glucan, water dikinase, and branching enzyme determination of starch structure. And for me, I want to appreciate the organizers for giving me this opportunity to make this presentation. And coincidentally for me, I will be doing the final presentation of my PhD thesis by this coming Friday. So it's just coming together for me, and it's just a wonderful opportunity. So I will go straight to talk about my project. So the mechanism for energy store is by glucose polymerization. And generally in animals, in yeast and bacteria, those levels of organism, the glucose polymer is called glycogen. And in plants, it is referred to as starch. And it is referred to as because of a lot of reasons, which I'm going to be explaining in subsequent slides. But Glycogen, keep in mind, is also a glucose polymer that is highly branched, and it is utilized as it is stored by all those organisms that I mentioned earlier on. So starch is an insoluble, non-structural carbohydrate, which is found in plants, mosses, fen, and microorganisms like algae and cyanobacteria. So, and starch is also important in plants as a metabolite which influences the growth of plants. So it's a major metabolite for plant with respect to growth. Then recently, starch has become a molecule, a key molecule, which mediates responses to abiotic stress, such as water deficit, high salinity, and extreme temperatures in plants. And that is just the relevance of starch to plants, the importance of starch to plant. But we as human beings, we utilize starch for different reasons. And the majority of the crop that is cultivated all over the world, wheat, rice, barley, potato, all of them, the major constituent of this crop is starch. And apart from the economic value of this crop, when we consume some of this crop as food, it comprises about 66% of our daily dietary calories, which means that it's a major energy supply for humans. Then apart from that, starch is also a renewable raw material for industries. Like in textile industry, the use of starch includes for sizing and for steaming textiles. Then in pharmaceutical companies, the use for the pharmaceutical industry, starch is used as SBEs because it has a slow release characteristics. Then, of course, bioenergy, which I'm sure many of us are familiar with. Starch is a major feedstock, and from which first generation bioethanol is derived. So I would like to talk about starch much more in details. So starch, the glucose polymer, after it is formed, it's formed into what is called granules. And when you look at that, the composition of that granules is made up of it's not just one glucose polymer this time around, but it's made up of amylose and amylopectin. And with respect to amylose is a linear polymer. It's a linear polymer. It's not really branched. But amylopectin is highly branched, and it's made up of about 80% of starch granules, though it varies from plant to plant composition. For some plants, amylose is just about 25%, while some it is about maybe 75% and what have you. And one of the things that makes starch very, very interesting to me is that apart from the fact that it is just made, the unit that makes up starch is just glucose, but the relatively simple chemical nature of starch is in stark contrast to the structural complexity of the final starch granule. As you can see, the pictures of the granules that I'm showing on my slide. And look at cassava. Cassava is a tropical plant that is grown in this part of the world in Africa. And potato, I'm sure we're familiar with that. Rice starch, wheat starch, and maize. And all of these plants, their starch, in terms of the granule structure, it varies with biological sources. And this has implications on the functionality of starch for industrial use, most importantly, and even for human consumption. And in terms of ratio of balance with respect to amylose and amylopectin, this has a strong implication in influencing the structural functionality of starch. For instance, a amylose-free wheat, which has been developed through, biotechnological means, is readily degradable by amylase and absorb and retain more water. So because of these characteristics, this characteristic increases the utility of this wheat, this amylose-free starch, both in formulation of animal feeds and in the production of biofuel. So this is very relevant to bioenergy. And additionally, apart from this amylose and amylopectin, which are the major component of starch granules, there's also a component of it which is the phosphate. That phosphate, even though it's in a very, very tiny amount, it also has to influence the specific functionalities of starch, and that will be seen in subsequent slide. So starch is limited because even though it's a good, industrial raw material, starch is in itself, in its native form, that is in its raw form, lacks the flexibility to function sufficiently, which means that it requires sometimes physical approaches to modify it. Sometimes it requires a chemical approach, while sometimes it requires the enzymatical approach to modify it. But more importantly, what is more relevant to me is a biotechnological approach for modification of starch. And it's important to emphasize that biotechnological modified starch come as a result of modification or alteration in starch biosynthetic enzyme. That is, there are several enzymes that are involved in the synthesis of starch. And I would like to describe very briefly the starch biosynthesis. Starch biosynthesis occurs through the reaction between water and carbon oxide in the presence of light. And once this occurs, glucose is produced. And after glucose is manufactured, the formation of granule formation now comes about with the concerted effort of several enzymes. And these enzymes play certain specific roles, and sometimes they play coordinated. They come together at times to also perform certain functions during starch biosynthesis. And according to its biological function, starch is categorized into two types: the transitory starch, which is the starch that is synthesized under daylight. And after the synthesis of that starch, the starch granule, it is stored in a starch storing cell called plastid in the leaves. In the night, this transitory starch is partly degraded by enzymes and transported in the form of sugars, mainly sucrose, to other parts of the plant. Some of these sugars are recovered, they are reconverted into starch and stored in amyloplast, another starch storing cell present in the seeds, tuber, and roots of eye plant. And this is called the storage starch. With respect to their biological function, transitory starch is used within 24 hours. But the storage starch is stored over a long period of time, and it is utilized mainly by the seeds after it is planted, or by tubers when it is planted. Initially, before the plant has the capacity to photosynthesize, the emerging plant, the seedlings, utilizes the energy that is stored in form of starch for its metabolic activities. So here is the pathway of starch synthesis, and that's what I'm showing on my screen. And glucose polymerization the starch synthesis starts with the synthesis of ADP glucose, which we can see down below there. And the enzyme that catalyzes that reaction is called ADP glucose pyrophosphorylase. Then after that, synthase is the enzyme responsible for the elongation of amylopectin chain. And this elongation, this ADP glucose is used as a glucosyl donor for this elongation to occur. So I will demonstrate with this next slide. So this is the demonstration of the schematic diagram of how this happens. And another form of enzyme that also perform the elongation in amylopectin this time around is called starch synthase. And that also does the work of elongation during amylopectin synthesis. And furthermore, another enzyme does the work of branching. The name of that enzyme is called starch branching enzyme. And that we could, after the branching of amylopectin, another enzyme called glucan water dikinase had starch covalently bound phosphate to the structure that is made. So it attached phosphate to the starch structure, amylopectin in particular. So next enzyme that also perform a very important work job during starch biosynthesis is isoamylase. And isoamylase actually does the work of trimming chains to ensure unsuitable starch granule crystalline structure. So what it happens is that wrongly placed glucan chains are trimmed by this isoamylase. The enzyme does the work of trimming down the glucan chain that are wrongly positioned so that it can enhance crystalline structure of starch. It can enhance the production of crystalline structure of starch. So although much is known about starch synthesis, there is still a lack of knowledge about many aspects. Many things are yet, we are yet to know many things about starch synthesis. And for example, it has through various, in previous work, there are indications that some of these starch biosynthetic enzymes interact with each other. They either interact physically or functionally. And in order for us to be able to really understand this level of interactions among these enzymes, we thought of repressing some of the genes encoding these enzymes simultaneously and to check the effect on the starch structure. And here is the example of some of the papers that have already been published with respect to interactions between enzymes in different plant, potato, maize, wheat, and what have you. So I chose potato for quite some reasons. Potato, number one, potato is the fourth, as I've mentioned, is fourth in rank in terms of production globally. Then starch, the potato tuber is made up of about 70% starch. The rest is consists of very little amount of protein, lipids, fibers, and a large amount of moisture. Then potato starch is also very high in food content, and this confers specific industrial function on it. Then the fourth reason why I selected potato is because it's very amenable to genetic engineering. Because using conventional method is kind of challenging because potato is highly heterozygous. It's has self-incompatibility, it has narrow genetic base, and it has acute inbreeding depression. So for that reason, traditional breeding is not so encouraged in improving potato crop. Then the public availability of potato also is one of the reasons why, of the genome of potato is one of the reasons why I selected the plant. So the background for this research is that starch branching enzyme, which I've earlier mentioned as the enzyme that causes the branching of amylopectin. In the previous research, it has been established that repression of starch branching enzyme, which has two isoforms, has some level of disparity in terms of the starch structure that is manufactured, but they also have some level of relationship. When starch branching enzyme 1 was repressed, there was no change in amylose content, there was limited change in the physical properties of starch. But when starch branching enzyme 2 was repressed, there was significant increase in amylose content, then limited change in physical properties. But when starch branching enzyme 1 and starch branching enzyme 2 were repressed simultaneously, there was about 90% increase in amylose content and change in the physical property of starch. And one common thing that is common to all of this research was that the starch-bound phosphate increased significantly in the 3 studies. So this leads us to the aim of the research, which is to assess if GWD1, starch branching enzyme 1 and starch branching enzyme 2 actually interact with each other during starch biosynthesis. Then the objective is to manufacture transgenic potato in which GWD1 and all of these isoforms are repressed singularly and in different combinations with each other. So I just want you to understand that. I try to understand that as much as possible. So, and we use RNA interference to silence those genes. And I will use this this image that I'm showing on my screen to explain the construction of this particular plasmid. We have about 6 plasmids that were constructed, but I selected one of it to use as an example. So in developing our plasmid, which was going to be incorporated, which was going to be used to, alter this gene in potato, we went through a series of stages to arrive at the point of developing the plasmid. Start with development of the donor vector. I mean, getting all of those the segment of GWD1 starch branching enzyme 2 into donor vector. Then we combine it in donor vector. We combine that in donor vector. Then in entry vector, an adapter which is going to help in transfer of this particular segment into the destination vector was added in entry vector, which is topo, and in destination vector, which is actually the plasmid that was used to transform potato. Well, these were the result that we got for all of those things, all of those different experiments that we conducted, and we could see that the band shows our DNA ladder is in hundreds. So the combination, if you had the number of base pairs that were, combined together for each of the experiments, it's about 600. So for each of all of these experiments, we were able to ascertain the insertion that we were actually targeting, which is going to be used to repress those genes in those targeted genes in potato. And this was a destination, this was a map of the destination vector. And if we look at the far right, there's a segment that is CCDB and another section that is CCDB. So we try to, incorporate our own created chimeric construct into this destination vector. And after we got our plasmid, then we use the plasmid to transform the agrobacterium, which is a bacteria that is actually used in engineering transformation in plants. So we see agrobacterium cell by the left and plant cell by the right. Through various combinations, when they come together of these two cells, there is a transfer of tDNA, within which is our the construct which we made, and that is conveyed into the plant cell and integrated into the nucleus in order for it to be able to carry out the things that we want it to do. So after we did that, within that tDNA, there's also a gene. There's also a gene that is actually, when it is expressed, it causes resistance to kanamycin. So we included kanamycin in our media, and we planted after the going through the transformation and the callus system and the shoot emerged. Then these are potatoes that I use this as just to describe. This symbolizes potato plant, and the shoots were planted in the media that contained kanamycin, and we could see that those that were transgenic actually grew roots in the media, whereas those that were not successfully transformed did not grow roots. So we discarded those that did not grow roots in the media, while we selected those that had roots in the media. Then we also check using PCR experiment to check for the presence of our insert in our putative transgenic consisting of the earlier mentioned construct. So here is the result of our findings. After we had gone through the transformation experiment, we were able to select, we had about 8 plants, putative plants that we selected, and from there we selected 2 per construct. So we selected 2 plants per construct to perform further analysis on them. And in the image that is shown by my left, we could see the reverse transcriptase PCR reaction showing repression of targeted gene for each of the transgenic lines that we have. And on the right as well, we try to quantify the amount of protein, GWD protein specifically. We raised through an immunoblotting, we raised an antibody to determine the accumulation of GWD within those transgenic lines, and we were able to establish that indeed there was reduction in the amount of GWD in constructs in which we were expecting it to be repressed. Unfortunately, the last one was starch branching enzyme 1, was the construct that was supposed to combine starch branching enzyme 1, starch branching enzyme 2, and GWD1 together. Due to COVID-19 issue, we were not able to get a transgenic line that was able to replace all of these three genes. So we discarded it and we moved on to our main, we moved further with the transgenic lines that we have. So after planting in the greenhouse and harvesting our tubers, we determined the phosphate content of the various transgenic line, and we find out that there was indeed increase in the phosphate content for the transgenic lines that has starch branching enzyme 1, starch branching enzyme 2, and the likes. Those were consistent with previous literature. For GWD1 as well, the work has been done, and there was reduction in the phosphate content, the starch phosphate content for that particular transgenic line, starch from that transgenic line. Now, for another transgenic line, which is starch branching enzyme 1 and GWD1 repressed lines, we're able to, see reduction in phosphate content as well. But it was not as much as what was established in GWD. And we thought that the reason for this may likely be that our immunoblotting result was not sensitive enough to detect the remnants of GWD protein, because GWD1 is the only enzyme that actually phosphorylates starch at that particular section, at that particular glucose point, point on the glucose. So, so we thought about another reason why this could be, and we said that it may be as a result of repressing starch branching enzyme alongside GWD1, which may be that the polyglucan that is synthesized act as a better substrate for the remaining GWD1 protein, unlike what was observed for GWD1 single transgenic line. So this is a plant. These are the transgenic line in comparison with the white type. And this picture was taken after 10 weeks of the experiment, and we realized that there was no much difference in terms of their physiological appearance. So, and at the same time, the phosphates, another point that I would like to quickly establish is that starch phosphorylation is the first step in starch degradation. And which means that glucan water dikinase is not only involved in adding phosphate to starch, it's also involved in starch degradation. So we test for starch degradation in leaf, and we were able to, after covering the plant for about 2 days with foil in the midst of darkness, we were able to ascertain that lines in which GWD1 were repressed, starch accumulated in those leaves for about 72 hours. Whereas those that GWD1 was not replaced, there was complete degradation of GWD1, the complete degradation of starch in those leaves. So the result on the analysis of tubers for all the transgenic line did not show any significant difference with respect to the tuber weight, with respect to the starch that was, that accumulated in the tubers, and even the structure, shape of the tubers were not altered in any way. So the analysis of tuber, so the starch that was taken, that we extracted from tubers, we did the analysis especially for the amylose content, which is a very important component of starch granules. And we find out that there was significant increase in the amylose content of starch branching enzyme 1 GWD1 transgenic line of potato. And the reason for this is that and of course, it has been established before in previous research also that lines in which GWD1 are repressed in potato, when the phosphate content is lower than 2 nanomole phosphate per mole exos equivalent, that amylose content increased in those lines. And that was very, very similar to the result that we obtained in our research. And the reason why for the difference between starch branching enzyme 1 GWD1 line and starch branching enzyme 2 GWD1 line, in terms of their amylose content, as it is shown in the figure above, is that we suspected that it is more difficult to explain this actually, but we thought that it may represent the difference between the starch branching isozymes in terms of how they branch non-phosphorylated glucan. And also, we also test the starch we also check the starch for the swelling power. The swelling power of starch was, we tried to determine the swelling power of the starch that we extracted, and we discovered that the swelling power of the starch for transgenic lines that were low in GWD1 lines, that were low in phosphate content, with the starch, the swelling power was lower in those lines that GWD1 was repressed, whereas those in which GWD1 was not repressed, they were increased. There was no commensurate. We couldn't, compare. There was no significant difference in terms of having higher phosphate content and the swelling power of the starch that was synthesized. So we also look for the freeze-thaw stability of the starch that was synthesized by those transgenic lines. And we also discovered that the freeze-thaw stability of those starch that in which GWD were repressed were more stable. They were more stable with respect to their freeze-thaw, rather more than those in which GWD were not repressed. And previous literature, it was established that there were other factors like the amylose content structure and amylopectin structure that affect the freeze-thaw stability of starch. And in that particular study by Joblin in 2002, it was actually emphasized that the reason why that occurred was because of amylopectin structure that was altered. But in our research, we find that it was not the case, that actually the phosphate content was actually what confers the resistance to freeze-thaw stability upon those starch. So we went further to check the granule structure of our various transgenic lines, the starch that was extracted from them, and it was only starch that were extracted for starch branching enzyme 1, starch branching enzyme 2 lines, and those that were established from starch branching enzyme 1 line that actually showed modification in terms of their granule structure. And we thought that the reason why it is so is simply because of the packing of amylose and amylopectin were affected as a result of repressing starch branching enzyme 1 and GWD1, or mainly as a result of combining starch branching enzyme 1 with the rest of the other two enzymes. Because we look at other two enzymes alone, there was no alteration in the granule structure. So we thought that the reason why there was alteration in the starch granules is simply because starch branching enzyme 1s were repressed alongside either starch branching enzyme 2 or GWD1. And another very important thing that is considered in potato is used in producing potato chips, and I'm sure that we know potato chips and French fries. And potato is usually stored under 4 degrees Celsius. When potato is stored, potato accumulates what is called reducing sugar. In that temperature, it accumulates reducing sugar. This reducing sugar actually, when potato is taken out and is being processed for production of French fries, it's this soluble sugar that has accumulated over that period of time in which it was stored reacts with free fatty acid, and it causes pigmentation on the products. At the same time, it also causes accumulation of acrylamide, which is very, very toxic. So we were thinking that if there's a possibility of reducing the accumulation of this in potato, that the issue with cold-induced stress will be taken care of. And that was what we did in our experiment. And we were able to ascertain that GWD1 was actually GWD1, and when repressed alongside starch branching enzyme 1 or starch branching enzyme 2, that it causes reduction in terms of the accumulation of soluble sugars during storage of tubers. And we were also able to establish this. This is also very important in the sense that starch branching enzyme 1 is known to be involved only in starch biosynthesis. But previous studies by Yando, Nelson, et al. They were able to, establish that in transgenic line, where starch branching enzyme 1 was repressed, there was a reduction in degradation of starch in the leaf of that cereal, specifically maize. But in this case, repression of starch branching enzyme 1 or starch branching enzyme 2 did not lead to reduction in terms of the starch degradation in starch. Rather, it actually just function in the same way that it will happen in the white type. So this in general, I'm concluding my work now so that I know I've taken most of our time. So during starch biosynthesis, starch branching enzyme 1 and GWD1 engages in synergistic interaction. And this was demonstrated by increased apparent amylose above either of starch branching enzyme 1 or GWD1 repressed lines. And as a result of this, novel starch with properties such as swelling power, freeze-thaw stability, and granule morphology were produced through this experiment. Lastly, the implication of this in the industry is that the starch is suitable for specific industrial application. So that is the end of my presentation, and I would like to acknowledge these people. These are the people that we shared the lab together while I was doing my research. And thank you very much. I will entertain questions at this point. Thank you very much.
Thank you, Muya, for your very good presentation. And from now, the questions and discussions session is open. So feel free to ask your questions and comments. We'll ask you to use the raise your hand function of Zoom to ask your questions, or write directly your questions in the chat box, and we'll go over them. Okay, so maybe I can start with one question.
Okay.
At the beginning, you said that there were 2 types of starch in
Yes.
in the different vegetables, like potatoes, and there was a transitory starch and the storage starch, from where
Yes.
the transitory starch is degraded for the then the synthesis of the storage starch. But is there a structural difference between the two, or some properties that are different between the transitory and the storage starch?
Really, thank you very much for the question. There are not too much differences in terms of the structure. Only that in Arabidopsis, a lot of work, most of this work is done in Arabidopsis. And we find out that actually that the amylose content in the granules that was extracted from the leaf of Arabidopsis were not as much as it would have been normally when you compare with the, what's it called, tissue, other tissues like tubers and the likes. But really, there were not too much difference, even in terms of structure, in terms of, there were not too many difference regarding that yet.
Okay, so technically, we could take the complete organism for the starch extraction without having any problem with, for example, the transitory starch would have lesser properties, and we would need higher level of purification. So we don't have that problem.
No, we don't have problem. But another thing that makes this study very important is that another that makes starch very interesting is the fact that this enzyme, for instance, thymic apamide starch synthase, which was this enzyme that actually does the elongation at the initial process, has about 5 isozymes or isoforms, and each of those isoforms performs specific function during such synthesis. And one thing that makes it very interesting is that some of these isoforms are highly synthesized, in plant, while they are not as their synthesis is less in other organs of the of plants. So we have that experience in plant. For instance, we know starch branching enzyme 2 is highly expressed in the leaf of potato, But when you check the potato tubers, it is not so highly, expressed. You don't see that much. You can't quantify it that much. But when you look at starch branching enzyme 1, it's highly expressed in the tubers, but not as highly expressed in the plant. So we have all of these variations, and all of this has a way of affecting the starch structure in the hen. So repression of one of these can lead to a particular phenotype in plant, in the leaf. Meanwhile, when you check it either in the tubers or in the endosperm, you tend to see something that is relatively different. So that's one of the things that actually make starch very interesting, and the research on starch is ongoing, on understanding the entire starch metabolism. So.
Also, I had a second question. When you show the results of the amount of reducing sugars on the different strains of the different mutant strains of potatoes, you show that GWD1 alone showed a reduction of degradation of starch, so there was lesser amounts of reducing sugars. But when I looked at the GWD1 and 2, there was more degradation. So how do you explain the difference between the two?
Okay, that there was more, sorry, let me get you. There was more degradation between GWD1, the transgenic line of GWD1, and starch branching enzyme 1 GWD line. Is that what you're saying?
Yeah,
okay, maybe I should go to the slide.
Yeah, you can go directly in the slide.
Okay. Sorry. Okay.
I do not see it yet. Okay. Yeah, if yeah, on the lower left for GWD1 and 2, we can see that in orange we have a lot more fructose than on the blue one. So is it more degradation?
Yeah, I get you very well.
Okay.
Sorry, I didn't mention during the presentation that it's this accumulation of reducing sugar only occur when potato tuber is stored under low temperature, when it's as low as 4 degrees Celsius. When it is at room temperature, it's almost normal in terms of how it accumulates. It's not as they are level at a degree at which it accumulates. This reducing sugar is not as much. So we use that as control. We use the keeping this potato tuber under low temperature, and we use storing it as room temperature as a control to compare the rate at which starch is degraded within those periods. And that was the result that we got. Actually, it was that low simply because glucose and fructose are actually the reducing sugars. And the reason why you have them so low is simply because there was reduction in starch degradation during this process, which leads to the lower accumulation of those sugars that you saw, that I presented. So if you see you will see it in all the transgenic line that the amount of fructose and glucose were almost as the same as the white type. I don't know whether that answers your question.
Yeah, it was more on if we compare the orange bar of GWD1-1 and GWD1-2, we can see that there is an increase between the orange bar with the C and the orange bar with the D, if I'm not mistaken.
Yeah. Each of the bar, like I said earlier on, the blue bar represents under what's it called, the room temperature, when it has room temperature, while when it is in what's it called, when it is in under cold stress, it represents the it's represented by the orange color. It's represented by the orange color, the bar. So the reason why there is a bit different, so we are comparing, so in this figure, you only compare those that are orange colors, not blue colors. So those are the things, those are the ones that you compare together. Why those that are blue are compared together. So basically, it's about in terms with respect to the amount of glucose, the fructose and glucose that was accumulated in those under those conditions. So that was the reason why there's that variation. I don't know whether you get what I'm trying to explain.
Yeah. Is there other questions? I don't think that we have other questions. So, so that conclude the activity. And I would like to thank everyone who joined and participated in the activity. And most importantly, thank you, Muya, for your time and presentation. And as a quick reminder, if anyone would like to present their work or an article, Muya, do you want to say something?
No,
okay.
Sorry.
Okay.
No.
So if anyone would like to present their work or an article, you can always register as a presenter on our website. Otherwise, you can always register as an attendee. And noteworthy, the next presentation will not be on August, but on September 26th for estival vacation. And we'll announce the next presentation presenter the second week of that month and on the website. So goodbye, everyone, and have a nice day, and see you next time.